Hi, everyone. My name is Marie, and I am a student at Trent University in Canada. And today I'll be talking to you all about the comparative genomics work that I've done on one of the world's smallest mammals. So some of you might have seen this video before, and for those who haven't, this is a shrew conga line, which is basically when a female shrew is trying to move her offspring from one location to another. They kind of all hold on to each other and move around, and it's very cute. But basically, this is one of the kind of interesting and unique traits that shrews have. Overall, shrews are actually one of the largest mammalian families on Earth. There's about over 400 known species all across the world. And obviously, with all these different species and different environments, some of them have kind of adapted differently to their different environments. So for example, the water shrew can actually dive underwater for up to a minute to kind of hide from predators or catch prey. The short-tailed shrew has a venomous saliva to paralyze its prey. And a few shrew species are actually known to echolocate. And overall, shrews are the smallest, shortest-lived mammals, and they have an extremely, extremely fast metabolic rate, which can actually become an issue because they can constantly have to eat basically every hour or so. And for shrews that live in regions where the winters are harsh and there's not a lot of food, this becomes an issue, right? So obviously, some of these species have had to adapt. And what has been seen in some shrew species is that in the wintertime, their body size actually shrinks. So that's like their organs, their skulls, their brains will physically shrink to kind of lower the energetical demands. And in the springtime, when resources kind of become available again, they actually will regrow, which is a pretty unique thing, and their brain actually regrows in their skull. And this is known as Donnell's phenomenon. And some studies on metabolites have also been have been done throughout the seasons to kind of see if there are any shifts. And a new study came out showing that there might be some shift from lipid to glucose as a source of fuel throughout the different seasons for shrews. So obviously a lot of very interesting things that are happening in shrews, which is kind of why I find them so interesting and what led me to wonder how do these special traits end up arising. So I decided to look at the DNA to kind of answer this question. And the idea here, my goal, was to find shrew-specific genomic variations that could maybe be associated to some of their kind of unique phenotypes. So what we did here is we looked at genomes, so full DNA sequences of 4 different shrew species, 2 of which we actually assembled their genomes, so the maritime and the smoky shrew, against 16 other mammals, and try and find basically genomic variants that are shared within shrew species but not found in other mammals. So here I'll just give you a quick background of the different 4 shrew species that I looked at. The first one being the common shrew that is very widespread throughout Europe and parts of Russia and a bit of Asia. Then the Etruscan shrew, also known as the Etruscan pygmy shrew, and it's actually the smallest shrew on Earth. It's extremely tiny and is found a bit in Europe again, in Asia, and a bit of Africa. The smoky shrew that is found around here, actually, in Montreal, basically eastern Canada and a bit of the United States. And finally, the maritime shrew that is actually one of the few endemic mammals found in Canada, and it's only found in Nova Scotia and in New Brunswick. So to do my research, I looked at 3 kind of main things. The first one being accelerated regions. So when we're looking at genomes, there are these regions that are very, very highly conserved and really haven't evolved much over time throughout the different species. So there's basically not a lot of mutations happening there. So if you're comparing, for example, the genome of a mouse versus a human, these regions would be very similar. And they're thought to be functionally important. So if we're thinking of regions involved with the embryonic development, for example, it would be very conserved throughout the different species because any mutations could kind of be very detrimental. However, sometimes these conserved regions do experience mutations, and they're called accelerated regions, and they're thought to contribute to species-specific traits. So for example, in elephants, this has been associated to cancer resistance, or in humans, accelerated regions have been found to be associated to kind of our higher cognitive abilities. So here the idea is that finding accelerated regions that are kind of unique in shrews might be associated to some of these shrew-specific traits that we're seeing. So we ended up finding 600,000 conserved regions. And if we're looking at the figure here, basically, the kind of outer ring just represents the different chromosomes. This was all mapped to the human genome. And the inner ring represents the locations of the shared accelerated regions in shrews. So any kind of yellow or golden line represents accelerated regions that were found in all 4 shrew species, and we found 35. And any blue line represents shared accelerated regions within 3 of the shrew species, or the Sorex kind of genus species that we have, so the maritime, common, and smoky shrew. And obviously we want to know these accelerated regions in what genes they're found. So many of them are actually found in genes associated to the nervous system development, to olfactory receptors, metabolism, and also echolocation. And actually, we found shared ARs in 2 shrew species for echolocation, and one of them is the common shrew, which is known to be able to echolocate. And then we also found these in the maritime shrew, but not a lot of work has been done on this species, so we don't actually know if it has the ability to echolocate or not, but it would make sense. It is more closely related to the common shrew, and many other Sorex species are known to be able to echolocate. So then the second thing that I kind of looked at was gene family size evolution. So when we're thinking of gene families, basically genes are considered as being in the same family if they come from the same ancestors. So whether it's through gene duplication or speciation, they're all considered as being in the same gene family. So here our question was, what gene families are experienced duplication events, and how many of these duplication events are happening? And here duplications are important in the context of adaptation and evolution because it just provides new genetic material for drift or mutation or selection to act upon, right? So new phenotypes can arise and new gene functions. So here, if we have rapidly expanding kind of gene families in shrews that are unique to those, they might be associated again to some of the unique phenotypes that we see. So we ended up identifying 2,000 to 3,000 gene duplication events in each shrew species, and we then looked at what gene families those duplications were associated with. So here we just have a little tree with all the species we looked at, and basically the first number that you see is the number of rapidly expanding gene families in each species, and then the number of rapidly contracting gene families in each species. And overall, we found that all the shrews shared 52 gene families that were rapidly expanding. And again, when kind of looking at what they were associated to, we saw nervous system developmental genes, genes associated to cellular response to stress and starvation, and then again, olfactory receptors. And then the last thing we decided to look at was positive selection using the DNDS rate ratio, so the amount of non-synonymous mutations over the amount of synonymous mutations. So quick recap. Synonymous mutations are mutations that don't change the amino acid sequence, whereas non-synonymous mutations do. So basically, by just dividing in the gene the amount of non-synonymous mutations that we see versus synonymous, we can get a DNDS value. And depending on what it is, this can kind of inform us of what's happening in this gene. So a DNDS of around 1 is basically the gene is evolving neutrally. Under 1, it appears like it's undergoing negative selection, and if a DNDS is over 1, then this is positive selection, and this variant could be advantageous. And here we're more kind of interested in any genes that are under positive selection because it is a driving force behind adaptive evolution and can kind of inform us on how shrews have been evolving and adapting over time. So this is basically all of the different genes that we looked at and kind of mapped again to the human chromosome to just see their location. And a lot of them, the genes that kind of came up as being positively selected were related to GTPases pathways, which doesn't mean much, but when we took a bit, we dug a bit more into these, we saw that they're associated to neuron development, neuron degradation, synaptic plasticity, and again, metabolic homeostasis. So here my goal initially, right, was to find shoe-specific genomic variations that could be associated to some of their distinctive phenotypes. So in all of the analyses that we did, we kept on finding stuff that was associated to the nervous system development, right? So this might be related to Donnell's phenomenon and maybe indicative of kind of the mechanisms that are happening that allow shoes to have this brain plasticity. And then we also found in each analysis genes that were related to metabolism, right? And we know that shoes have a very, very fast metabolic rate and that they might be undergoing these kind of metabolic shifts throughout the winter. So overall, the shoe-specific kind of variations that we found seem to maybe be associated to some of the distinctive phenotypes that we see, and specifically kind of related to their survival throughout the winter. And obviously a lot more work needs to be done to really link all those traits and basically what's happening in the genome, but now at least we have a list of many genes that we could kind of look a bit more into to get more information. So thanks all for listening, and I hope that now I convinced you all that shrews are very interesting little mammals. Yep.
So you said you mapped the shrew genome to the human
Yep.
Reference genome.
Yeah.
Why did you choose the human reference genome? What's the closest related mammal?
No, so basically when we looked at all 20 species, it made it easier for all the different analyses that we have because we had some files basically of all the other 16 mammals that we looked at. Basically mapping everything takes a very long time. So some other researchers had already done that, all mapped to the human. So what we did was we kind of took the species that we were missing, did that, and then that helped us a lot. And also just for visualization purposes, it's easier to visualize 22, 23 chromosomes than 1,000 sometimes, depending on the assembly, so that's why.
Thank you.
Well, thank you, everyone.
Okay, hello and welcome to my talk. I am very excited to talk to you about population genomics of wild avian embryos and how we think we uncovered some hidden genetic diversity. And I think most of you can appreciate that genomic variation is not just about SNPs, even though a lot of population genomic studies work with SNPs and also with samples in adults. But actually, there can be many different types of genomic variation, and we are interested, for example, in variation that happens between different developmental stages. And in songbirds, we actually have a case of such variation, the germline restricted chromosome, which occurs in germ cells and gets repulsed or expulsed in somatic cells. And there's already some things known about this GRC. So apparently it's there in males with 1 copy, but in females with 2 copies. Most studies have been done on zebra finches, so we don't know so much about how it differs between different species. And we also don't know precisely when and how the GRC is expulsed during the development. So we wanted to study this, and our study organism is the great tit. And the karyotype in the great tit seems to be quite representative because most songbird species have the same karyotype. The great tit also has a ZW system of sex determination, where the female is the heterogametic sex, even though in our reference assembly we don't have the W chromosome yet. And we also know that the GRC of the great tit is quite large. I don't know, can you see the cursor? No. Yeah, it's not very visible, but otherwise you could see that it has the same size as some of the largest chromosomes in the karyotype of the great tit. So how could we go about finding if there is such genomic variation? We think we can do it like this. So if you imagine here on the left side, you have alleles from a position in an autosome that map to the reference at the bottom. But then additionally, you have some other sequence that is homologous and also maps to this region. And if they are close enough to map to the same region, but a little bit different, you will have a different ratio of the alleles, and it will appear as heterozygote, even though it's not. And also the number of reads that map to this region will be different from the surrounding regions. So our hypothesis was that the GRC or the W chromosome could be mapping to autosomal regions and appear as heterozygote. And we then predicted that sites with excess heterozygosity that come through this process should have a higher sequencing depth, and also that there should be differences between males and females and or between embryos and adults. So to study that, we sequenced some great tit eggs. We took the DNA from embryos of 14 great tit eggs from a population in Germany, in Bielefeld, here in the middle. There we had 7 males and 7 females. And we also compared it with sequences from a previously published study that had sampled the blood of 10 adult great tits from across Europe. These are the sampling sites. And both these sets of samples were Illumina sequenced. And then I did whole genome population genomic analysis with angst. In particular, I looked at the differences between the eggs from Bielefeld and the adults from across Europe, and also whether the male eggs and female eggs within that Bielefeld site differed. And the first thing I did was just simply comparing the genetic diversity between eggs and adults. And this was actually quite surprising to us. So here on the x-axis is the diversity in the eggs, on the y-axis the diversity in the adults, and 3 different diversity measures. And for all of them, the eggs always have higher genetic diversity in all the larger chromosomes and also the linkage groups and so on, which is really surprising because the eggs come from just one sampling site in Germany and the adults come from all across Europe. So why would the eggs have a higher diversity? So in order to find that out, I did an allele frequency spectrum where you plot the number of times an allele occurs in the sample and then the proportion of SNPs that fall into that category. I have 10 adults in blue and 10 eggs in orange, and the eggs are half male, half female. And you can see in the eggs we have a slight excess of fixed variants here at the 10, but also an excess of alleles with a frequency of 5. And then we thought this can only be due to the 5 females in the sample. So in order to confirm that, I looked at the heterozygosity between female eggs and male eggs. And you can see, especially on the chromosomes at the top, that in the lower right corner of each subplot, there are these regions where the female eggs have an excess heterozygosity compared to the male eggs. So could this now be such a structural variation that just looks like it's a heterozygosity? And it seems like it does. So here, if you look at the sites that are heterozygous in almost all individuals, here 0.9 and 1, for example, on the right-hand side of the graphs, they have also a very high sequencing depth, which is exactly what you expect if additional sequences would map there and then would make it look like it's heterozygous. So then we were wondering where exactly in the genome this happens. And I did an FST analysis between male and female eggs. The red line is the global FST value, and I color-coded the regions where the FST is more than twice as high as the global average in red. And you can see here for chromosome 1 and 8 that there are really distinct regions where the males and females are very differentiated. And here in chromosome 8, there's even one really large region. Overall, I identified 50 such regions, almost all chromosomes. And if you add them up together, it's about 6.7% of the total genome. So then I picked some size-matched regions on the same chromosomes as a control region for these sexually differentiated regions. And again, we find that in the sexually differentiated regions, here in red, they have a higher sequencing depth than in the control regions. And this effect is even stronger in females. It's not so easily visible in the graph, but if I test it, this interaction effect is significant. And then a colleague of mine did an analysis of the repeats in these regions of sexually differentiation. And this was really surprising to us. So apparently 60% of these regions are segmental duplications. And at the same time, these regions are also enriched in genes compared to the control region. So it seems like most of it is segmental duplications, and in the rest of it, there are just a lot of genes with a very high density there. So then I did an enrichment analysis of these sexually differentiated regions. And actually, the thing that stands out is that these genes were enriched in annotation terms that are related to reproduction, especially female reproduction, while in the control region, these annotation terms were not enriched. So what can we conclude from this? We think there's some sort of genomic dark matter that we sequence, and then doing the mapping, it maps to the autosomes. And this is what we are picking up. The embryo specificity suggests that it could maybe be this germline restricted chromosome. The sex specificity could also mean that it's the W chromosome, which is not yet in the reference genome. But whatever which of these it is, we think it would be good if we try to assemble it. So we are doing nanopore sequencing at the moment and try to really get some longer scaffolds or context to actually see what this genomic dark matter is. We're also now considering how widespread this phenomenon is. Is it restricted to the Bielefeld population in Germany only? Do other European populations have this, or even other songbird species? And even if we're not completely sure yet what it is, I think it's still important that we take these regions into account, because you can imagine if you do population genomic studies and you don't think about this, and maybe in one sample you have more males than in the other, then you would think these populations are different, even though it's just a sex effect from these weird regions. Yes, and with that, I would like to thank my PI, Tony, Justin Wilcox, who did this repeat analysis, Quentin Foucault, who maintained the field site and sampled the eggs, and Yuqi Chen, who did the mapping of the sequences. And I thank you for your attention, and I'm happy to take questions now. Yes.
And so you suggested that GRC has an effect, that it has some sort of role in the divergence that you saw. I was just curious, because it's just in the germline, do you think you sample enough cells in your sequencing to have the signal of genes from the GRC? Which is related to the second part of the question. I know that they found that there was enrichment for certain types of genes associated with female reproduction in the GRC in blue ticks.
Mm-hmm.
So I wondered if you actually made a comparison between the blue tick GRC genes and then what you're seeing as the signals in the autosomes, because we know that these genes all have paralogs elsewhere in the genome on the GRC. So I'm just curious if you...
Yeah, we haven't done that yet. Yeah, one reason is also because we know that apparently the gene content of the GRC can also change really fast. So I don't know if it actually, the GRC of the blue tit actually has the same set of genes as the gray tit. But yeah, I think we will look at this in the future. Yeah. What was the first part of your question?
Well, it was just, do you actually sample enough of these?
Yeah, this is a good question. So the reason why we came up with this idea is because we see this effect, and it seems to be specific to the developmental stage. And then we thought, oh, it could be the GRC. But you wouldn't, from the beginning, think that, or it wouldn't be your first idea, because, as you say, probably not all the cells have the GRC, even in the embryo. So we're still a little bit puzzled about this, yeah. I think there was another question.
I was just wondering about the consistency of coverage for the females versus males. It's not that you just have like one female that's a bit bonkers and she's pulling up the average, like are all the females at higher coverage?
No, they all have about the same coverage of 40x. And yeah, for some analysis, I also took the mean sequencing depth over all males versus the mean sequencing depth of the other sex. So I don't think there's...
So I was asking within sex if there's much variation in coverage.
Yeah, I don't think they are all between like 40x or 45x. It's all... I mean, when we send it to the sequencing center, we tell them it should all be the same. Of course, there's some variation, but I think they're quite similar.
Okay. Hi, everyone. My name is Emily Kerns. I'm a PhD candidate in Jesse Weber's lab at the University of Wisconsin-Madison. And thank you all for coming to learn a little bit about what I've been working on, looking at how commonly used sequencing methods can capture variation in DNA methylation in 3' stickleback. So phenotypic plasticity is ubiquitous in nature. These are just a few of my favorite examples, from squirrels altering the size of their litter based on environmental cues, to Daphnia growing extra body armor in the presence of predators, and lizard populations in urban heat islands having a higher thermal maximum than nearby forest populations, largely due to the evolution of phenotypic plasticity. But to understand how plasticity evolves, we need to understand the mechanisms that underlie the ability to mount a plastic response. So the mechanism I'll talk to you guys about today is DNA methylation. DNA methylation is an epigenetic marker. It is the addition of a methyl group to cytosine, and in vertebrates, it's typically found at CpG sites. Methylation often facilitates the formation of heterochromatin, and it can block transcription factors from binding to DNA, so it's typically associated with reduced transcription. It's also largely a heritable trait, although it's also environmentally sensitive in that methyl marks can be added or removed to the genome in response to environmental stimulus. And so we often talk about methylation as a binary, presence or absence, but when we do bulk sequencing, we're sequencing many cells, so we can also have intermediately methylated CpG sites. And this is introduced both by variation within cell types and variation between cell types. And so this is relevant for something that I'll talk about later, but in order to understand how methylation can contribute to evolution, we need to be able to quantify the variation in methylation due to ancestry versus the variation due to environment. And the three-spine stickleback is an excellent organism to study this. There's an ancestrally marine population that has undergone an adaptive radiation into freshwater, and there's a growing body of literature that suggests methylation may have helped facilitate some of these freshwater adaptations. So there's a class of sequencing methods called bisulfite sequencing that people often use to look at DNA methylation. So unmethylated cytosines are converted to uracil during a bisulfite conversion reaction. And then during library preparation, those uracils are converted to thymine. So you can compare your sample to a reference genome, and CT mismatches are inferred to be unmethylated cytosines. And this can be done on the whole genome, but whole genome sequencing is often expensive, so we often have to do have lower sample sizes and typically have a lower read depth. And additionally, many of the reads that are mapped with whole genome bisulfite sequencing lack these CpG sites and are therefore uninformative for methylation. Alternatively, we can do reduced representation bisulfite sequencing, and this is very similar to RADseq in that an enzyme is used to target specific regions of the genome. The most commonly used enzyme is MSP1, so we can target CpG-rich regions of the genome and sequence a much smaller proportion of the genome at a greater read depth. But the huge discrepancy between the information that we get from these two sequencing methods led me to ask, how do they vary in their ability to quantify evolutionarily meaningful CpG sites? So to address this, I also went back to our fundamental question of looking at variation due to ancestry versus variation due to environment. So if we were to look at this in PCA space, we might find, if we have multiple populations that were reared in multiple environments, we might find no clustering by population or environment. We could find what I'm calling the ancestry first outcome, and this is where the greatest axis of variation along PC1 separates individuals based on their population or their ancestry. And we could find potentially a secondary axis of variation based on their rearing environment, or we could find no separation based on environment. And then finally, we could have the opposite, the environment first outcome, in which we find the greatest axis of variation is determined by rearing environment, and again, potentially a secondary axis based on ancestry, or no separation based on ancestry. So based on previous stickleback research looking at DNA methylation, I predicted we would find the ancestry first outcome. So to address these questions, I sampled 5 stickleback populations, and I have individuals from all populations from a common garden, and we also sampled 4 of these populations in the wild. So we can ask about heritable variation versus plastic variation based on the rearing environment. I also have 2 different evolutionary axes of divergence represented here. I have a benthic ecotype and a limnetic ecotype, which are commonly studied in stickleback. And I also have a population that is very resistant to a common tapeworm versus a population that is tolerant to tapeworm infection. And so, in addition, I had biological replicates from the common garden that I used for whole genome bisulfite sequencing. And specifically looking at the benthic limnetic axis, I'm also looking at these 2 ecotypes for other projects, and I have a broader prediction that because benthic fish have evolved in more variable habitats, they would be more plastic than limnetic fish. So to analyze my data, I used TrimGalore for adapter trimming. I used BWA-meth for read mapping. This uses the BWA-mem algorithm, but it does in silico bisulfite conversion of the reference genome to increase mapping efficiency. And then I used methyl-DACL to tabulate per CpG methylation metrics and for SNP filtering. And then I did statistical analysis and data visualization using methylkit and ggplot and R. So this is the whole genome, one of the whole genome samples. So I'm going to show you guys a biological replicate first. And the x-axis in this figure is the % methylation per base, and the y-axis is the frequency. And so the bin all the way on the left are CpG sites that are always unmethylated. The bin all the way on the right are CpG sites that are always methylated. And everything in the middle are those intermediately methylated sites. And so the biggest pattern that was apparent to us looking at our biological replicates was that we lost a lot of these intermediately methylated sites. But there were a couple different studies that came out in the last year that suggested that these may not be informative sites for evolution. One found that they are not reliably transmitted during cell division and are typically enriched at genes that are not expressed or very lowly expressed. And another study found that there's greater nucleotide diversity at the genome in regions where these intermediately methylated sites are found, suggesting perhaps there's relaxed selection in these regions of the genome and less regulation maintaining methylation state. So what I did was I took all of my RRBS data and I put it in a PCA, and I did not find any evidence for the ancestry first or the environment first hypothesis. But to look more into these intermediately methylated sites, I separated those from stably methylated sites, with the prediction that we might find evidence for the ancestry first outcome with the stable methylation, and again, no evidence of this using just the intermediately methylated sites. But again, I did not find anything. So I went back to my original axes of divergence and asking about variation due to heritability versus plasticity. And so first looking at the benthic versus limnetic ecotype, I did actually find evidence of this ancestry first hypothesis, and I found 20 differentially methylated sites. So if you're not familiar, this is a volcano plot. The x-axis is the magnitude of difference in methylation, and the y-axis is the negative log of the Q value. So anything above that line are significant sites. And again, I found the same pattern looking at the tolerant versus the resistant population. And in this comparison, I found 7 differentially methylated sites. And then I looked within each population and compared fish caught in the wild versus those reared in the common garden to ask about plastic methylation. And I found variable amounts of plastic methylation in each population, although I did want to highlight, I did find more differentially methylated sites in the benthic population than the limnetic population, which is in line with my more general prediction. But I found all the way from 2 to 61 plastic differentially methylated cytosines in all of these populations. And so I wanted to ask if any of these plastic sites were shared between populations. And so I did find, this is the same plastic DMCs, just in a Venn diagram, and I did find one shared plastic site between the benthic and the limnetic population. Although most of the plastic sites are population specific, which is in line with previous literature that suggests that the plastic response to methylation depends on the pre-existing context of the methylation. So this was in line with previous results. So in review, we did find a reduction in intermediately methylated sites using the reduced representation method, but RRBS still enriches for those CpG-rich regions and also results in a greater read depth, so potentially greater accuracy in methylation calls. And I do encourage people studying methylation to continue to look into those intermediately methylated sites and ask if they are informative for evolutionary questions. And we also found evidence of the ancestry first hypothesis, but only when we looked at previously identified evolutionary axes of divergence. So this really emphasizes the need to be careful about what comparisons we're making so that we're not masking real evolutionary signal with noise. And finally, in line with my prediction, the benthic population did have more plastic DMCs than the limnetic population, and I'm happy to talk more with anybody about my other projects looking at plastic variation between the benthic and limnetic ecotypes after my talk. And so thank you all to my lab, and I'm happy to take any questions if I have time. I do.
So what kind of function are linked to the CPG site?
Yeah, so the question is if there's any functional link of that one plastic CPG site that was shared between populations. And I have not yet done functional annotations yet. So that is kind of like my next step in this analysis. So I'll let you know. Yeah.
Have you looked at the concordance between just two methods to see if you get the same proportion of methylated sites at the same sites, and might the read depth difference in the methods contribute to the amount of intermediate methylated sites you see?
Yeah, that's a great question. So asking if I find the same differentially methylated sites between whole genome versus reduced representation, and if I've taken read depth into account in the methylation calls. I have attempted. I only have 2 samples from 2 different populations for the whole genome data, but I have not looked at how read depth has influenced the methylation calls. So that's a great suggestion. Thank you.
Hello, everyone. Maybe I can start now. So I'm a PhD student at Max Planck Institute for Plant Breeding Research, but I'm working on animals. I'm working on frog, and since our lab is focusing on polyploidy animals and plants. And today I will presenting some preliminary results regarding our long-term ongoing research, sex chromosome evolution on this Australian boring frog near Batrachus. So polyploidy is not that common in is not that common in animals compared to plants. Almost all mammals and birds, they maintain stable diploids, but this is not absolutely. Especially some clades, for example, amphibians and fish, maybe because they don't need undergo dosage decomposition, or they don't have a degenerated sex chromosome, so they still can have a lot of polyploid. Then what is polyploid? So traditionally, polyploidy can be divided into 2 types, autopolyploids and allopolyploids. Autopolyploids, which means it originates within one species. So after genome doubling, they are not diverged. They don't pair preferentially. So they usually still form tetravalent and has ring structures. Meanwhile, allopolyploid, it means it originating after hybridization between species, and they are already diverged within 2 subgenome and with disomic inheritance. And everything in between, introduced by Stebbins in 1950, called segmental polyploidy, which means some fragment of chromosome it can show tetrasomic inheritance mode, meanwhile the rest can show disomic inheritance. We call it a mixed inheritance. Why we pick up frogs, since frogs can have both types, both allo and auto. And also, we think maybe because most of them have homomorphic sex chromosome, which means 2 chromosomes, they can't distinguish by ions. They look the same. They just have a sex determination region. They don't have the entire sex chromosome. So maybe because of this, they could tolerate to be polyploidy more easier. And also, maybe you can you already noticed that from this tree, frogs they can frequently occur. Transitions can within one species, can between different species, can among the entire clade. So this yellow arrow means the transitions. So far, I think the only well-studied frog is in Xylopus family. Both diploid and tetraploid are there, but tetraploid Xylopus levis is allopolyploid, so it's from 2 diploid species. And this highlight small red box means is our frog near butcher holes. The reason why we choose this frog genus since regarding our unpublished paper, we think we already figured out there are 2 auto polyploids and 1 potential allo polyploid with a mixed inheritance mode. The rest 6 species in blue, they are diploid species. And with this model, we already sequenced a population level whole genome resequencing data based on museum samples. Since these frogs, they are burrowing frog, their reproduction is affected by precipitation. So it's, we don't really know the generation time of them, and it's not that easy to capture them. So that's why we working with museum samples. So with this data set and this model, so there are 2 straightforward questions in our hand. First, we want to figure out the self-determination modes in the genus level. And then after quantify this in every species, we want to know the effect of allo and auto on sex determination and sex chromosome evolution. So let's start from diploid. So far, we already assembled this diploid, and we managed to publish it in our preprint paper. So we use 10x plus high C technology, and we got a chromosome level genome with genome size 2.1 gigabyte, and N50 is over 227 MB. But this is just a diploid, so tetraploid should be, the genome size should be double. But we haven't assembled it yet. So lastly, I selected samples from my population data, which I know sex information. So I got 9 male and equal female samples. I mapped to this genome, and I treat male and female as 2 subpopulations, calculate SNP-based FST between them. So theoretically, if there's a potential sex determination region or sexually differentiated region, it should be abnormal peak when we plot it along the genome. So as the Manhattan plot show here, you can see on the left, we find 2 peaks in 2 different chromosomes. The first peak, there are in total 5 genes, but these 4 genes, they are located in the conserved region in all vertebrate. And especially in the DMIT gene family, they are usually play a role in sex determination in birds, in some fish and frogs. Meanwhile, the second region is pretty large. It spans 10 megabytes across the chromosome and involved in 112 genes. Since so many genes are there, we are very curious about if there are some certain categories or terms that are overrepresented in this region. So we performed the gene ontology analysis in this region. The result is shown on the right. So interesting, these terms, they're indeed related to sex. So especially we are interested about this term, fusum. Fusum originally found in the insect is a structure, but later researchers also found it in frogs, Xylopus levis, and this fusum structure, it indeed play different role in males and females. So after we find these 2 potential sex determination regions, naturally we want to figure out sex determination systems in these 2 regions. So first, I checked the highest FST region in the chromosome 1. So here plot on the top left is, I just zoom in chromosome 1 of the Manhattan plot. You can see within this peak, these 5 genes, I could find 20 SNPs. There seems to sex-linked SNPs because they are always heterozygous in all male samples. Meanwhile, this sites, they are always homozygous in all female samples. And also, these 20 SNPs, we checked the linkage disequilibrium among them. So the hidden member is showing in the bottom left. So this diagram color, if they are deep, means their LD relationship is very strong. So compare these 2 results, we think it might be a XY system since these SNPs, they are always heterozygous in males. So the same approach I applied on the second peak, and so you can see this peak, they're pretty large, and I can only find a few SNPs. Here is 7 SNPs. They are still behave like they are always heterozygous in males. In females this time. Meanwhile, this position is always homozygous in all male samples. But you can see this physical position of these 7 SNPs, which you can see on the top right. They are pretty far away with each other, so we don't think they have a strong LD. So now we think the second region should be the sexually differentiated region, but the first region, the first peak I just mentioned, it should be the sex determining region. But so they are physically unlinked, but they are functionally linked with each other. But how they interact with each other, we need more data and to address this question. So far, what I'm talking about is focusing on this diploid species in this yellow circle. So since I still don't have non-reads for polyploid species, instead of waiting for upcoming non-reads, so I map my tetraploid samples onto my diploid species, and then use a same approach. I just calculate genetic diversity between tetraploid males and female samples, and then plot them onto diploid genome. You can see the Manhattan plot on the left is very noisy, but since we already have 2 candidate peak we got from diploid pictals, so we just focus on these 2 category regions. So interesting, the first peak, it still could be found in tetraploid pseudola, but the second peak on chromosome 10, the pattern, this non-block is disappeared in tetraploid. So we think maybe tetraploid have new patterns and new scenarios, or maybe it's just because of we don't have tetraploid genome and our sample size of tetraploid is not enough to get these differences between males and females. So let's go back to 2 questions I said. So far still at the first phase, and specifically I just figured out one diploid species sex determination system, I think. And for this tetraploid, we think it at least shared the first peak with diploid species, but the rest we have no idea. So summary is that, so we find 2 peaks in diploid pictals. The first peak we think is XY, and it's very small, just 5 genes are there. The second peak, according to sex-linked SNPs, it might be a ZW, but if it's ZW, how the segregation with the first peak at meiosis, we don't know. And we don't think it's sex determination region, the second peak. We think it's just maybe driven by selection. But how it interacts with the first peak, we don't know. So then the tetraploid, since I mapped them onto diploid species genome and I plotted it along diploid genome, I could find these genes on first peak. So we think it should share the first sex determination region with diploid species, but the rest I don't know. So still it's a lot of things to do after I can quantify the sex determination patterns in the genus. Then thanks for my advisor Polina Novikova, who gives me this super interesting project. I very like this model; it's very interesting and unique. And also thanks to our collaborators from Australia. Without them, we couldn't reach this population level data. So and the rest of people in our lab. Thank you, everyone. Okay, thank you. Okay, okay.
So you only looked at like, is it 6 individuals of each sex?
Um,
Six or how many individuals of each sex when you were looking for?
for diploid, here I use is 9 male, 9 females. Yeah.
I was just wondering about the smaller region that you have there. I mean, is there any chance it could just be something random because you just have 9 individuals written and 7 SNPs, or you say that it's pretty, like you have a haplotype block that's quite long?
Yeah. The first pick with,
The one with the females, the one that's where you think might be ZW.
so the first pick we think we already knew it because the pattern, according to certainly analysis with other vertebrates, especially birds, so we think it should be a cellular mining region. Yeah. But the second pick, yes. So we actually, maybe this is more straightforward. We think maybe it's an inversion or structural variation here. It's just driven by selection. It's not the cellular mining region.
That's what I was just wondering. Could it possibly be some kind of
Yeah.
artifact of the genome, something that
Yeah, exactly. Yeah.
understand what's going on there?